This lesson will be about an amazing story, how a lifeless rock turned into a rock teeming with life. Of course, Earth is our home, but the human species is just one of millions that currently inhabit this planet. Now, in the mid-1800s, Charles Darwin introduces a powerful way to understand the history of life on Earth, and that is the tree of life. Now, why does the tree of life have so many branches? Well, we've learned that there are many ways to make a living on Earth, so natural selection will tend to multiply species over time. Now, this is a picture of a poster, actually, that illustrates the tree of life. The tips of the tree are currently existing species. So, for example, we've got some mammals up here, and as we go down a branch, we're moving back in time, back towards extinct ancestors. And so the branching nature of the tree means that species multiply from common ancestors. In fact, you can just say any animal and any plant will have a common ancestor. So we just go down the plant branch, down the animal branch until they converge. It just so happens that the common ancestor of plants and animals is a single-celled organism, of course, which looks neither like a plant or an animal. And at the very base of the tree, the root of the tree, will be the origin of life itself. Darwin also speculated about the origin of life. You'll recall that when several species share a trait, Darwin's going to consider the possibility that they inherited the shared trait from a common ancestor in the past. Well, since all living things are composed of cells, Darwin inferred that the first living thing, too, was a simple cell, perhaps assembled in a warm little pond long ago. Now, how does one study the origin of life? Well, it's helpful to consider the conditions on Earth when life might have arisen. We're talking about nearly 4 billion years ago, and what scientists think happened was that life emerged from geochemistry. That is, chemistry on the early Earth produced important molecules that later assembled into the first living cells. So this suggests a research strategy. Try to simulate the conditions of the early Earth, and then you want to study the kinds of molecules produced by different chemical processes. In 1952, Stanley Miller did just that, and he's going to open up a whole new field of research into the origin of life. He's going to try to simulate the early Earth with this apparatus here. He's got a chamber here. He puts water in it to simulate the oceans. Apply a little heat and you'll get evaporation like you find on the Earth. He's got another chamber here where he will introduce various gases that he thought were present in the atmosphere. Gases like nitrogen, hydrogen, methane, ammonia, and then the water vapor. He'll introduce a spark here to simulate lightning, and he hopes that some interesting chemistry will be going on here. Whatever new molecules produced will be collected down in this fluid here. Now, the fluid initially, the water, was clear. After a couple days, the water kind of turned brown, and he analyzed the chemicals found in that water, and he found very interesting molecules, amino acids, the building blocks of proteins, and lots of other organic compounds. These are molecules that have carbon in them. So these are lots of the kinds of molecules you find inside cells. Now, he did not create a living cell, but he did manage to synthesize many of the molecules found inside cells. And this excited lots of people at the time. And so a whole new branch of science was, in a sense, created by Miller and his colleagues, those interested in studying the origin of life. Now, there were some criticisms. Later, NASA scientists identified the atmospheric gases on planets like Mars and Venus. Now, Earth likely had a similar atmosphere early on. And based on what they discovered, Miller's mixture was not realistic. He got the gas mixture wrong here. When you use more realistic gases, you don't get as much of the interesting chemistry. You don't get as many of the amino acids and other organic compounds. In addition, molecules would have drifted away from each other in such a vast solution as the ocean. See, the idea here is if this chemistry is going on in the atmosphere and then the interesting new chemicals are raining down into the ocean is huge. How could those molecules be concentrated enough to then assemble into the first living cells? So there were some major criticisms of this early result. So the search continued. Are there places on Earth where chemistry might occur and concentrate the resulting organic molecules? Well, in the 70s, some really interesting rocky structures were found at the bottom of the oceans. They're called hydrothermal vents, and this one is an alkaline hydrothermal vent. Now, These hydrothermal vents are regions of volcanic activity on the ocean floor. And the vent has a constant supply of energy and simple molecules from the Earth's interior. And in addition, importantly, there are very small chambers throughout the vent capable of concentrating organic molecules so that further chemistry can happen. In fact, the minerals in the rocky walls of the compartments speed up chemical reactions. So it began to look like these vents were a really interesting place where chemistry might have been going on in the early Earth. Was it a possible solution to some of the problems with Miller's approach? So here we see sort of a diagram of one of these vents. We've got these rocky compartments that are magnified here. So this is the magnification of one of the compartments. We've got simple atoms, simple molecules coming up from the Earth's interior that are going to be entering these chambers here. We've got the walls of the rocky compartments have certain minerals that can facilitate chemistry going on here. So we're calling this geochemistry. We've got a source of energy and raw materials percolating up through these rocky chambers. We've got a situation where whatever molecules produced by chemistry are going to be concentrated by these small little chambers. Perhaps really interesting chemistry went on here on the early Earth. Now, the product of that interesting chemistry was a complex arrangement of molecules that we call a living cell. Cells are like modern cars. They have a boundary, an engine, and a computer. And all cells have these three basic functional parts. So like a car, a cell has a boundary. It defines the self versus the environment. Like a car, a cell has an engine responsible for powering the operation of the cell. And like a car, a cell has a computer. And the computer will control the operations going on inside the cell. If we use the biological terms, then cells have a membrane that confines the cell's contents to a small volume so that important molecules can interact. So the boundary of the cell is the cell membrane. Cytoplasm, a watery fluid, fills the interior of the cell where proteins perform the chemistry of life. So here we see the engine of the cell is a bunch of these molecules called proteins, and they perform lots of different chemical reactions in the cell. Some of those chemical reactions are extracting energy from food or the environment in some way, and some of those chemical reactions are building other parts of the cell. So this would be the metabolic engine of the cell. And DNA then stores the information to make the cell's proteins. So DNA is acting like the computer of the cell. It has the information to build these important protein parts. We can think of proteins as the workers of the cell, performing lots of different jobs in the cell. And DNA then would be management. The DNA not only has the information, the recipes to build these parts, but the DNA controls when these parts are made and how much of each part is made, and generally controls the chemistry going on inside the cell. Let's break it down farther then. The membrane is composed of molecules called lipids. A lipid molecule is composed of atoms of carbon and oxygen and hydrogen and phosphorus. The tails of the lipid here are a bunch of carbon atoms linked together with hydrogens stuck to them. The proteins are composed of building blocks called amino acids. Notice the amino acids are built from carbon atoms, nitrogen, hydrogen, oxygen, and this would be one amino acid, and a protein would be a chain molecule, a bunch of amino acids stuck together. Here are some cartoon proteins here. And then the DNA molecule, a very complex molecule composed of hydrogen atoms and oxygen and nitrogen and carbon and phosphorus. And this molecule has three amazing capacities. It can store information, the information to build another molecule, the proteins, the workers of the cell. It can be duplicated, so DNA as a molecule can be duplicated, which allows for a living thing to reproduce, pass on this DNA molecule to its offspring. But when the DNA is duplicated, we'll see sometimes mistakes happen. We call them mutations. So this DNA can change, and that leads to variation in offspring in subsequent generations. Now, later in the course, we'll use this diagram to sort of work our way through the chemistry going on inside of a cell. This would be a simple bacteria cell, but a simple cell is really a complex arrangement of molecules, lots of different molecules interacting in many different kinds of ways. But the net result of that complex interaction is the whole cell is alive. So cells are composed of molecules, which are themselves composed of atoms. And that brings us back to our story in the universe, that the atoms made by stars, ejected into space by supernova, chemically bonded to form simple molecules in the dust of nebulae, which later swirled around a new star and built large particles and rocks under the influence of electrostatic and gravitational forces, ultimately assembled into a planet with water on its surface and a great diversity of chemical reactions, which after hundreds of millions of years produce the molecules with the capacity to make a living cell. Now on the right here, we see the familiar Drake equation, and the N sub E term is the number of habitable worlds in one solar system. Of course, we said the Earth is in the habitable zone, so liquid water exists on the surface. And water is so important, we think, for life because it's great at facilitating chemistry. And you need chemistry to build complex molecules. Again, if we take a look at the cell in the middle of this diagram and break down the parts of the cell, the membrane is made of lipids, and lipids are complex molecules. Glucose is a sugar. That's one of the energy molecules that lots of cells use. Amino acids stuck together in long chains make complex molecules called proteins. And DNA is a very complex molecule. Importantly, these molecules, built from atoms, are not alive. Yet the entire cell is alive. This means that the emergence of life on Earth is an example of what scientists call spontaneous generation. Let's go back to our Drake equation. The next term in the equation, after N sub E, is the fraction of the habitable worlds where life emerges. Well, we just said living things were made of molecules. Cells are made of molecules, and molecules are not alive. So if non-living molecules assembled to form a living cell, that's an example of spontaneous generation, life from non-living stuff. Now, this is an old idea. For thousands of years, people thought that non-living stuff like dirt or dead organic matter could spontaneously generate living things. And it took a while to test that idea and reject it. So today, scientists do not think living things can spontaneously generate. Rather, all living things today come from pre-existing living things. But the one big exception would be the origin of life. That's an example of spontaneous generation. A living cell is made of non-living molecules. And in fact, that then would apply to all living things in our galaxy or the universe. Living things, if they're made from non-living atoms and molecules, living things are spontaneously generating. In the bottom part of the diagram, we see one possible scenario for the spontaneous generation of life here on Earth, and that would be the undersea volcanic vents, these hydrothermal vents. And after hundreds of millions of years of chemistry producing complex molecules which assemble into a living structure, it escapes its rocky nursery and goes on to colonize the world. If that's the case, we really do live on one strange rock. Our planet Earth is a bunch of rock and metal and water and an atmosphere, but that planet produced life. Life grew out of our planet. Now, if we step back, how does a planet produce life? Well, you take some stardust and you put it in some water in a small compartment. So you got a pot. You add some heat so that powers the chemistry, and you get some very complicated molecules that organize into life. What were the chances? That, of course, is the big question. This F sub L term. What is the fraction of habitable worlds where life emerges? It is a hugely important question to modern science. And remember, just what is it that's emerging? Life, the self-sustaining chemical system capable of Darwinian evolution. Now, there are many questions that remain about the origin of life. Here are two important ones. How were the key molecules synthesized? How and where did this take place on the Earth? Again, some scientists think underneath the oceans in these hydrothermal vents. And then how did the genetic code emerge? How was it that this kind of molecule, this DNA type of molecule, or its close cousin, RNA, came to store information about building another molecule, the proteins? Very, very difficult question to address, but scientists are actively studying this question. Still, though, while it may seem impossible that this kind of thing could have emerged, we have to remember the timescales. We're talking about 300 million years of experimental chemistry. So if you give the Earth 300 million years to do lots of different kinds of chemistry, it may well be that you can produce very complicated molecules that assemble into a structure that we call living.